Hello everyone and welcome to the first video of the biology playlist of Minute MYP. In today's video, we're going to be covering mitosis. So, what is mitosis? Mitosis is a type of cell division where one parent cell it divides into two identical daughter cells. Two identical daughter cells, and the keyword here is that the daughter cells are identical. And what this means in terms of chromosomes is that there's something called a diploid division that's happening. So, as you know, each cell in our body, each somatic body cell, is going to have 23 pairs of chromosomes, which means 46 chromosomes in total. What happens here is that if the parent has 46 chromosomes, each of the daughter cells will also have 46 chromosomes. And these daughter cells will both be genetically identical to each other and to the parent cell. So, this is an example of the diploid division. My bad, I think I spelled that wrong. This is an example of a diploid division where the same number of chromosomes are there in the parent cell and the daughter cells. So the functions of mitosis are growth, because without your cells dividing in this way, you won't be able to grow taller, grow larger body parts, and grow in general. Because that's how any organism increases in size and becomes bigger, because its cells divide by mitosis. Then you have repair, because your new skin cells, for example, if you have a cut, form not because they're dropped from the sky, because they divide and they form a new skin layer. So the cell division into multiple new skin cells by mitosis is what causes your cut to heal over with the new layer of skin. And finally, we have asexual reproduction. So now we have the definition and the functions of mitosis nailed down. Now we're going to go to the phases of mitosis. I like to remember the four phases of mitosis as being PMAT. So if we start with the first phase, it's called prophase. Prophase is where we have, let's say I'll make a cell drawing here, and let's say we have the nucleus here, and we have the chromosomes. I'm representing them by an X's right now. You have this. What happens in prophase is that these chromosomes condense and become visible. So that's the first part. This just means that they become thicker. So let's say I'll make all of these chromosomes a little bit thicker. So in prophase, all the chromosomes condense and become more visible. What also happens is the nuclear membrane, this blue thing, starts breaking down around the chromosomes. So this just breaks down and disappears. So now we just have these chromosomes which are condensed. And now also what happens here is that something called spindle fibers are formed. Spindle fibers are essentially you can say ropes that move the that move the chromosomes to different parts of the cells for division in the later stages of PMAT. So these spindle fibers are originating from a organelle called a centriole. So you can say this is a centriole, this is a centriole, and then there's spindle fibers that are coming out of these centrioles. That's what you have in prophase. Now, if we go to M stands for metaphase. In metaphase, we have the chromosomes, the condensed chromosomes lining up in the equator of the cell. So if you imagine just a line down the middle of the cell over here, all of these chromosomes right here, they're going to just be now lining up in a row in the center, on the equator of the cell. And then the spindle fibers that we have, now I'll just erase this. The spindle fibers that we had earlier, they start attaching to a certain part of the chromosomes called the centromere. Now, just to break down what exactly centromere is, let me draw a slightly enlarged chromosome right here. Let's say this is our chromosome. As you can see, if you split this right into half, you get two halves. Each half is called a chromatid. So two chromatids make up a chromosome. A centromere is essentially, you can say, the joining point. This is where the two sister chromatids are held together to form a complete chromosome. So this point over here is called the centromere. Now, what happens is the spindle fibers that we were talking about earlier, they attach to the centromeres of each of the chromosomes. So let's say now that I've explained you where the centromere is, you can visualize these spindle fibers attaching to those centromeres on either side. Like so. Now we have the next phase called anaphase. Anaphase is essentially where these spindle fibers pull these chromatids apart from the centromere. So they were attached right now to the centromere, and now in anaphase they're going to basically pull those two sister chromatids apart. So it's going to separate the chromosome. So just pull it apart from the centromere with the help of those rope-like spindle fibers that you can say play tug of war with the chromosomes and just split them apart. So if I draw that, we're gonna get some spindle fibers on this end, some spindle fibers here, and we're going to have we're going to have a chromatid on each one. So we're going to have a chromatid, 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 and then we're going to have the other half right here. So if you can see what happened, earlier it was an X for the chromosome, and now it's become separated into two halves, and the spindle fibers pulled these two sister chromatids apart. So that's what basically happens in anaphase. Now if you visualize, my cell diagram is a little small, but if you see over here, they are now considered to be at opposite poles of the cell. They are now at opposite end of the cells. So in anaphase, the spindle fibers pull the sister chromatids apart and move them to the opposite poles of the cell. Finally, we have telophase. Telophase is essentially where you have this whole setup now with the chromatids at each opposite end, and we have the spindle fibers are now like not relevant anymore because they're at opposite ends of the cell over here. Let's say like this. Let's draw one more. So now we have this setup over here, and in telophase we have the chromosomes at the pole, and then remember the nuclear membrane that disappeared earlier. Now since we want two identical daughter cells, each one will be its own cell, so it'll each have a nucleus. So we'll have like a nuclear membrane forming around each pole. And now you can see where this is going. Two nuclear membranes means two cells are going to form, and a nuclear membrane around each end of the cell where the chromatids were. And now finally this is the end of PMAT, and all this leads to a process called cytokinesis. In cytokinesis, what happens is that the cell splits apart. Let me draw something like this. It basically splits apart here. If you can see, this is basically the cytoplasm of the cell, so it's pretty easy to split apart. What happens is, now that we have the new nuclear membranes, we'll have one here, and we're going to have our chromosomes right here. Now, if you can see what's happened, it's basically the cytoplasm is just splitting along a point called the cleavage furrow. So this point where the cell splits into two new cells at last is called the cleavage furrow. So you can just imagine that you're physically pulling like the jelly of the cell apart, and it's splitting into two individual yet identical pieces. So that's how PMAT and cytokinesis works in mitosis. And that's basically the rundown of mitosis for this video. Thank you.